Hello, friend. Welcome to Science for Sleep. Whether you have returned tonight or have only just discovered this channel, this is a quiet place to slow your thoughts, learn something gentle, and allow the day to fade little by little. Tonight, we are beginning with one of the most familiar vegetables in the world, a carrot. You can picture it resting in your hand. It may be 20 centimeters long, about the length of a dinner knife, with a broad shoulder near the top and a narrow tip below. Its skin is marked by faint horizontal lines and tiny pale scars, where fine side roots once reached into the soil. At the wide end is a rough circular crown. That is where a low cluster of green leaves once rose into the sunlight. We usually meet the carrot after it has been washed, trimmed, and separated from the rest of the plant. Yet the orange piece on a cutting board is only one organ from a larger living body. Its scientific name is Daucus carota, subspecies Sativus. It belongs to the Apiaceae family, along with parsley, celery, parsnip, fennel, coriander, and cumin. Many members of this family have finely divided leaves and small flowers arranged in umbrella-shaped clusters. The carrot's most famous feature remains below the ground. Before we look more closely, if you enjoy these long, quiet journeys through science, you are welcome to subscribe. And if it feels natural, you can leave a comment sharing where you are listening from tonight. It helps other curious listeners find this calm corner of the world. For now, you can simply settle in and let the story continue. The part we eat is mainly an enlarged taproot. A taproot is the first central root that grows from a young seedling. It travels downward, while smaller roots branch from its sides. This makes a carrot different from several other foods that also grow underground. A potato is a tuber, which is a swollen underground stem. An onion is a bulb made mostly from thick layers of modified leaves. Ginger is a rhizome, another kind of underground stem that grows sideways. A carrot is true root tissue, enlarged into a place where the plant can store water and food. If you cut one across the middle, you can often see two broad regions. The outer ring is usually darker, softer, and sweeter. It contains much of the tissue that carries sugars from the leaves and stores them in living cells. The center is often paler and firmer. It contains more xylem, the tissue that carried water and dissolved minerals upward from the soil. Near the outside is a thin protective skin. Beneath it are millions of living storage cells filled with water, sugars, pigments, and other compounds. The pointed lower end was once connected to the actively growing root tip. The small marks along the sides show where lateral roots emerged to gather more water and minerals. Even after harvest, the carrot cells remain alive for a time. They continue to use oxygen and release carbon dioxide as stored sugars are slowly broken down. This helps explain why an old carrot gradually loses moisture, sweetness, and crispness. The greatest surprise is that the root was never meant to be the plant's final achievement. Carrots are biennial plants. A biennial normally completes its natural life over 2 growing seasons. During the first season, the carrot forms a low rosette of leaves and sends much of the energy those leaves capture into the thickening root. Then winter arrives. If the plant survives the cold, it can use those underground reserves during the second year. A tall flowering stem rises from the crown, sometimes reaching about 1 meter in height. It opens many small white flowers and begins producing the next generation. People usually harvest the carrot before any of that happens. We take it at the end of its first season, while its stored energy is still concentrated underground. The familiar vegetable is an unfinished plant. It is a reserve gathered for a spring that most cultivated carrots will never reach. And before that reserve became thick, sweet, and orange, the entire future plant was folded inside something only a few millimeters long. A few of these tiny beginnings could rest easily in the center of a gardener's palm. They are grey-brown, dry, and uneven, with shapes closer to small splinters than the smooth seeds shown in simple drawings. A wild carrot unit is usually only about 2 to 4 millimeters long. Its width may be between 1.3 and 2.3 millimeters. Several could fit across the face of a fingernail. Nothing about them suggests the long orange root that may eventually form beneath the soil. Even the word seed is slightly misleading. The object sold in a packet as one carrot seed is usually a structure called a mericarp. A mericarp is 1/2 of a dry fruit. The complete fruit is known as a schizocarp, a word used for fruits that split into separate pieces when they mature. In a carrot, the schizocarp divides into 2 mericarps. Each half contains a single true seed. This means the small object placed into the soil carries both a thin layer of fruit tissue and the seed protected inside it. The outer surface has several raised ribs running from one end to the other. Fine bristles project from some of those ribs, giving the dry fruit a rough texture. On a wild plant, these bristles can catch against fur, feathers, clothing, or nearby vegetation. That contact can carry the fruit away from the parent plant before it falls. Commercial carrot seed often looks smoother. During processing, some of the longer hairs may be rubbed away so the pieces move more easily through planting equipment. Small oil ducts also run through the fruit wall. Plants in the carrot family often produce aromatic oils, which help give their leaves, fruits, and roots their recognizable scents. Beneath the outer layers sits the seed coat. Inside that coat lies a tiny embryo, already organized into the beginning of a root, and the first structures that will grow upward. Stored food surrounds or supports this embryo during its earliest growth. The entire young plant remains still while the mericarp is dry. Its cells are alive, though their activity has slowed to a very low level. Germination begins when the conditions around it become suitable. Water enters the dry tissues and causes them to swell. Enzymes become active again. Stored materials begin breaking down into forms the embryo can use. Oxygen in the small air spaces between soil particles supports respiration, the process that releases usable energy from stored food. Temperature affects how quickly these reactions proceed. Carrots can germinate in cool soil, although colder conditions usually make the process slower. The seed must also remain close enough to the surface for the young shoot to reach daylight. Growers commonly cover carrot seed with only a thin layer of soil. Its small size creates practical problems. One mericarp can be difficult to pick up between 2 fingertips. A light breeze can shift it. Water from a hose or heavy rain can move it into a small hollow. Several may fall together, even when the gardener tries to spread them evenly. Close spacing produces crowded seedlings that must later be thinned. For easier planting, seed companies may coat each mericarp in a round layer of clay or another harmless material. These pelleted seeds are larger, smoother, and easier to place at regular distances. Some growers use seed tape instead. The mericarps are fixed at measured intervals along a narrow strip that is laid into a shallow row and covered. Large farms rely on precision seeders designed to release these irregular units at controlled spacing. Carrots are usually planted directly where they will grow. A young taproot begins forming almost immediately after germination. Moving the seedling later can injure that root or bend its tip. The harvested carrot may then become forked, twisted, or shortened. Direct sowing allows the first root to enter undisturbed soil from the beginning. Patience matters during the days that follow. Radishes may appear above the soil in less than a week under good conditions. Carrots often take between 1 and 3 weeks. During that time, the row can look completely empty. The upper layer of soil must remain moist because the tiny seedling has very little stored water and cannot yet reach deeper reserves. A dry crust at the surface can stop emergence even after germination has begun below it. Under the soil, however, the stillness has already ended. The seed coat has opened. The embryo has resumed growth. Its first root is pressing outward into darkness, where gravity will begin guiding the direction of every new cell. Moisture has entered the dry tissues and awakened the embryo inside. The first structure to emerge is the radicle, the young plant's embryonic root. It breaks through the seed covering before the first leaves appear. This early order matters. A seedling needs a steady supply of water before it can build much tissue above the ground. The radicle begins that work in complete darkness. Its tip presses into a crowded world of mineral grains and living material. Some particles are coarse pieces of sand. Others are flat, microscopic pieces of clay that hold water against their surfaces. Between them lie fragments of dead leaves, fungal threads, bacteria, narrow air spaces, and thin films of moisture. The young root must move through this uneven mixture while its own tissues remain soft and easily damaged. A small protective structure covers the end. This is the root cap. Its outer cells absorb pressure and friction as the tip moves between soil particles. Some are scraped away during growth. New cells replace them from deeper inside the root cap, allowing the protective layer to renew itself. The root also releases slippery substances called mucilage. This clear material helps reduce friction and changes the narrow zone of soil touching the root. Just behind the cap lies the root's growing point. Biologists call this region the apical meristem. A meristem is a group of cells that can keep dividing to produce new plant tissue. The cells near the meristem remain small and tightly packed. As new cells are formed, older ones are pushed farther behind the tip. They then absorb water and become much longer. Most of the root's increase in length happens in this zone of elongation, rather than at the very point. The tip leads the way, while the tissue behind it stretches the root deeper. Direction comes from specialized cells inside the central part of the root cap. This region is called the columella. Its cells contain small structures known as amyloplasts. Amyloplasts store starch, which makes them denser than much of the material around them. Inside a gravity-sensing cell, they settle toward the lowest side. Researchers often call these starch-filled structures statoliths when discussing their role in gravity detection. Their movement gives the plant physical information about which side of the root faces downward. That information changes the distribution of auxin. Auxin is a plant hormone, meaning it is a chemical signal that affects growth and development. When a root lies at an angle, more auxin collects along its lower side. Root cells respond to auxin differently from stem cells. At the concentration found on the lower side, auxin slows cell elongation there. Cells along the upper side continue lengthening more quickly. Uneven growth gradually curves the tip downward. This response is called positive gravitropism. The word positive means that the organ grows toward the pull of gravity. A stem usually shows the opposite response as it bends upward toward open air and light. The root can correct its course again if a stone shifts it sideways. Its statoliths settle into a new position. Auxin distribution changes, and growth becomes uneven until the tip points downward once more. No muscles pull the root into place. No nerve carries a message through the seedling. The direction emerges from moving starch bodies, chemical signals, and differences in cell expansion. Farther behind the elongation zone, young cells begin taking on specialized jobs. Some become part of the water-conducting tissue. Others form the outer surface. Many epidermal cells produce root hairs. Each root hair is a narrow extension of one cell. It may look like a tiny side root, although it has no separate growing tip or internal vascular system. Thousands of these extensions spread through the microscopic spaces around soil particles. Their combined surface area allows the seedling to absorb far more water and dissolve minerals than a smooth root could collect. Root hairs are delicate and usually short-lived. New ones form closer to the advancing tip, as older ones farther back collapse or are rubbed away. The result is a moving zone of absorption that follows the root into fresh soil. By this stage, the young carrot has established its basic downward path. It is still pale and thread-like, with only a small amount of stored material. The long orange shape familiar from a kitchen remains far in the future. Length alone cannot create that form. The narrow root must soon begin adding tissue around its center, slowly changing its direction of growth from downward movement into outward expansion. The young root continues downward as a narrow, pale strand. It may already be several centimeters long, yet its width remains close to that of a thread. At this stage, most of its work involves reaching new soil and absorbing water through the root hairs behind its tip. There is very little room inside for storage. The change begins deeper within the root, around the tissues that carry water and sugar. A thin band of dividing cells gradually forms there. This band is called the vascular cambium. A cambium is a meristem, which means it contains cells able to divide repeatedly and produce new tissue. The vascular cambium eventually becomes a continuous cylinder running around the root center. Each new division adds cells on both sides of this narrow living ring. Cells produced toward the inside become secondary xylem. Xylem carries water and dissolved minerals upward from the roots. Cells produced toward the outside become secondary phloem. Phloem transports sugars and other organic materials from the leaves into growing or storage tissues. The word secondary refers to growth added after the root has already begun lengthening. Primary growth extends the root downward. Secondary growth increases its diameter. As these divisions continue, the root slowly changes from a thin absorbing structure into a broad storage organ. Many of the new cells become parenchyma cells. Parenchyma cells have relatively thin walls and remain alive after they mature. They can hold water, sugars, pigments, and other compounds inside large internal spaces called vacuoles. These storage cells make up much of the soft flesh found in a carrot. Their walls contain far less lignin than the cells in wood. Lignin is a hard material that strengthens tree trunks and older stems. A carrot needs enough strength to hold its shape in the soil, while remaining flexible enough to expand as more material arrives. The vascular cambium keeps adding tissue in a controlled pattern. New xylem accumulates closer to the center. Phloem and storage tissue develop farther outward. Cell enlargement adds even more width as water enters the vacuoles and gently stretches the walls. If a carrot is sliced across its middle, these different tissues can often be seen without a microscope. The center may form a paler, firmer circle. This inner region contains a larger share of xylem tissue. The outer flesh is often darker orange and slightly sweeter, because it usually contains more phloem and pigment-rich storage cells. The difference varies among cultivars. Some carrots have a small, tender core that nearly matches the surrounding tissue. Others develop a wider center with a lighter color and firmer texture. Breeders often select for roots with an even color and a less noticeable core, especially when the carrots are intended for fresh eating. Growth near the surface also requires protection. As the root widens, its first outer covering cannot stretch forever. A second meristem called the cork cambium begins producing protective cells near the surface. These cells help replace tissues that split or wear away as the diameter increases. The faint lines and rough patches on a mature carrot partly reflect this continuing renewal. The final width depends on several processes working together. Cambial cells must keep dividing. Storage cells need enough water to expand. Hormones coordinate growth between the root and the leaves. Genes influence how quickly the root thickens and whether its finished shape becomes long, blunt, round, or tapered. The surrounding soil also pushes back. Loose soil allows even expansion. Hard layers or stones can press against one side and distort the growing root. A carrot therefore becomes wider through thousands of small additions. Every new layer records another period of cell division and enlargement. The solid root seen on a cutting board is built from living tissues arranged over many weeks. By the time it reaches harvest size, millions of storage cells have formed around the root center. Those cells provide the space required for sweetness, color, and stored energy. Their walls create the container. The material that fills them must be produced above the ground, where a low crown of leaves is beginning to gather light. Above the widening root, a low circle of green leaves spreads across the soil. This leaf pattern is called a rosette. Each leaf is divided into many narrow segments, giving carrot foliage its soft, feathery appearance. The fine shape helps expose a large surface to light, while allowing air to move between the leaflets. Beneath that canopy, the thickening root remains hidden. Its growth depends on work taking place in the open air. Thousands of tiny pores called stomata cover the leaf surfaces. Each pore can open or close as two curved guard cells change shape around it. When a stoma opens, carbon dioxide enters from the atmosphere. At the same time, water vapor escapes from the moist tissues inside the leaf. The water being lost must be replaced. Fine roots absorb it from the soil, and xylem carries it upward through the crown and into the leaf veins. Inside the leaf cells are chloroplasts, small structures containing the green pigment chlorophyll. Chlorophyll absorbs energy from sunlight. That energy helps the plant combine carbon dioxide from the air with water drawn from the ground. The process is photosynthesis. Its products include energy-rich carbohydrates that the plant can use, transport, or store. The first sugars produced inside a leaf are quickly changed and reorganized. Much of the material sent over longer distances travels as sucrose. Sucrose is the same general kind of sugar found in ordinary table sugar. Although inside a plant, it serves as a mobile package of chemical energy and carbon. A mature carrot leaf can produce more sugar than it needs for its own immediate growth. Biologists describe such a leaf as a source. A source exports useful material to other parts of the plant. The enlarging root is a sink. A sink uses or stores more sugar than it produces locally. Young leaves can also act as sinks while they are still expanding. The growing root tip draws resources as well. The carrot plant must keep adjusting how much material reaches each destination. Sucrose enters narrow living tubes in the phloem. Loading sugar into these tubes increases the concentration of dissolved material inside them. Water then moves in from nearby xylem tissue. The added water raises pressure within the phloem. This pressure helps push sugar-rich sap away from the source leaf and toward tissues that are actively using or storing it. When the sap reaches the root, sucrose leaves the transport system. Some of it is broken down to release energy for cell division and repair. Another share becomes part of new cell walls, or is stored in the living parenchyma cells that formed around the vascular center. The root's sweet taste develops partly from the sugars held in these tissues. Carbon arriving from the leaves also contributes to pigments, proteins, and defensive chemicals. Water fills the large vacuoles inside storage cells and helps create the pressure that keeps those cells firm. A crisp carrot, therefore, depends on both the carbon captured above ground and the water absorbed below it. The flow changes as the plant ages. While the root is filling, it behaves mainly as a sink. If the carrot survives winter and begins flowering during its second year, stored reserves can move out of the root. At that stage, the root becomes a source for the rising stem and developing flowers. The direction of transport follows need. Growth also depends on balance. Nitrogen helps the plant build chlorophyll, enzymes, and new leaf tissue. A shortage can leave the foliage pale and slow. An excessive supply may encourage a large leafy canopy, while reducing the root qualities growers want. The plant may send more resources toward fresh top growth instead of steady storage below. Water supply, temperature, leaf damage, and day length also influence how much sugar reaches the root. A shaded plant captures less light. Leaves damaged by insects lose productive area. Drought can close the stomata, limiting both water loss and the entry of carbon dioxide. The carrot's hidden growth reflects conditions across the entire plant. Its orange body depends on a connected system stretching from the smallest root hair to the highest leaf tip. Sugar can arrive in abundance, yet the root still has to expand through the physical world around it. Every stone, hard layer, and crowded patch of soil can redirect that growth and leave a lasting mark on the shape below. The expanding root pushes against a world that is rarely uniform. A few centimeters below the surface, loose grains may give way to a hard band pressed down by feet, machinery, or repeated rain. Small stones interrupt the path. Old roots cross through the same space. Some pockets hold air, while others remain filled with water after a storm. The carrot responds to each condition as it grows. A straight root develops most easily in deep, loose soil with good drainage. Farmers often describe this kind of soil as friable, meaning it crumbles easily instead of forming hard blocks. Sandy loam is especially useful. It contains enough sand to leave open spaces, along with smaller particles and organic matter that hold moisture. The young tip can continue downward through this mixture, with fairly even resistance. At the same time, the storage tissues behind it can widen without being squeezed sharply from one side. Commercial carrots with long, narrow shapes may need 25 centimeters or more of workable soil. A hard layer at half that depth can stop the tip early. The root may become short and blunt, even when the leaves above appear healthy. A stone can bend the growing tip sideways. Compacted clay may slow one side more than the other, producing a curve. The most dramatic changes occur when the young central tip is injured. A healthy taproot usually maintains one dominant downward axis. Damage can weaken that control. Several side roots may then continue growing and begin storing food themselves. The result is a forked carrot with two or more thick branches. Transplanting can cause this kind of injury, which is one reason carrots are usually sown directly into their final bed. Fresh manure that has not fully broken down may also create uneven conditions and encourage excessive side root growth. Crowded soil can produce a dense covering of fine roots. A carrot growing in poorly drained ground may struggle to obtain enough oxygen. Roots need oxygen for respiration, the process that releases usable energy inside their cells. When water fills most of the air spaces in soil, oxygen moves through it very slowly. Growth weakens, tissues become stressed, and disease-causing organisms may gain an advantage. The root can remain small or develop a rough, hairy surface. Water supply leaves another kind of record. During a long dry period, cell expansion slows. The root continues living, though it adds new tissue more slowly and may become tougher. A sudden heavy watering can restart rapid expansion. Water enters the storage cells. Their internal pressure rises and the tissues swell. Sometimes the inside expands faster than the outer layers can stretch. A split opens along the length of the root. Similar cracking can occur when heavy rain follows several dry weeks. Steady moisture produces more even growth. Severe shortages can leave carrots undersized, irregular, or unusually firm. Stress may also increase bitter or harsh flavors, especially when heat accompanies drought. Spacing matters because every root needs its own volume of soil. Carrot seeds are so small that several often land close together. After emergence, growers remove extra seedlings in a process called thinning. The remaining plants may be left several centimeters apart, depending on the expected size of the cultivar. Without thinning, neighboring roots compete for water and minerals. Their leaves shade one another, reducing sugar production. Physical crowding also limits the space available for the storage roots to widen. Several carrots pressed together may become narrow, flattened, or twisted around one another. A misshapen root does not always come from a stone or dry soil. Microscopic roundworms called nematodes can damage growing tips and create forks or swollen areas. Fungal and water mold diseases can kill parts of a young root, forcing growth to continue from surviving side tissues. Those causes may remain hidden until harvest. Two carrots with similar shapes can therefore carry very different histories beneath their skin. One may have met a stone. Another may have suffered an infection during its first weeks of growth. A crooked carrot is a physical record of the space it occupied and the pressures it survived. Its bends mark changes in direction. Its cracks preserve sudden changes in water supply. Its narrow places show where expansion was limited. Shape develops from the root surroundings, while its internal quality keeps changing with age. As more days pass underground, the balance of water, sugar, fiber, aroma, and pigment continues to shift. A carrot can be pulled from the soil long before the plant has finished developing. For a gardener, the first usable harvest may begin when the root is only as wide as a finger. Its tissues are still growing. Its leaves are still sending sugar downward, and its cells remain active. The word mature usually means that the carrot has reached a useful size for the person growing it. Biology continues beyond that chosen moment. Young roots often contain a high proportion of water. Their cell walls are relatively tender, and the central core may still be narrow. A small carrot can feel crisp without being woody, with a mild flavor and little bitterness. Leaving it in the ground adds more mass. The vascular cambium continues producing tissue, storage cells expand, and the root's diameter increases. Extra time can deepen color and allow more sugar to accumulate. It can also bring a larger core, firmer fiber, stronger aromas, or a rougher texture. The outcome depends partly on the cultivar. A cultivar is a plant variety maintained because it has a useful set of inherited traits. Small, early carrots may be ready about 60 days after sowing. Many common garden varieties require roughly 70 to 80 days. Large roots bred for winter storage can take 100 days or longer. These numbers are estimates rather than fixed deadlines. A packet marked 70 days does not guarantee that every root will reach the same size on the 70th morning. Growth slows during cold weather. Cloudy weeks reduce the amount of sugar made by the leaves. Crowding limits light and soil space. Poor nutrition may delay enlargement, while excessive nitrogen can produce strong foliage without equally strong root growth. Heat creates a different set of pressures. Carrots are cool season crops, and prolonged hot conditions can reduce quality. High temperatures may strengthen bitter flavors or make the roots coarse. Warm soil can also dry quickly, slowing cell expansion unless water is replaced. Drought, followed by heavy rain, may cause rapid swelling and cracks. Constantly wet ground limits oxygen around the root and raises the chance of decay. The calendar therefore measures only part of the carrot's age. Temperature and water supply shape the pace at which those days are used. Two roots planted on the same morning may develop differently if one grows beside a shaded fence and the other receives full sun. Even carrots in a single row can vary because soil depth, spacing, and moisture change across short distances. Commercial growers need a narrower harvest window than a home gardener. A small garden can supply 1 carrot for dinner, while the rest remain underground. A farm may need thousands of roots to enter machinery on the same day. Harvest timing is based on several physical details. Diameter is one of the easiest to measure. Fresh market carrots must also have the desired length and taper, meaning the way the root narrows from shoulder to tip. Tip fill describes how fully the lower end has developed. A poorly filled tip may remain thin or pointed, when the market expects a smoother shape. Color should be strong and reasonably even. The tops may need enough strength to survive mechanical lifting. Roots grown for cut and peel processing face another requirement. Many products sold as baby carrots begin as full-size roots that are cut into short pieces and rounded. The original carrot must be long enough to produce several usable sections. Its width should remain consistent, so the finished pieces come out close to the same size. Harvesting too early wastes potential yield. Waiting too long can increase cracking, disease damage, core size, or toughness. Older roots may become more exposed to insects and soil pathogens simply because they remain in the field longer. A carrot never reaches one universal stage called ripe, in the way a soft fruit changes at the end of development. Its cells keep respiring. Sugars continue moving and changing. Protective compounds respond to weather and injury. Growers choose the point when size, texture, color, and storage ability fit the intended use. That chosen moment marks maturity for the harvest crew. Inside the plant, the root still serves a future purpose. Its stored reserves are waiting to support a tall, flowering stem after winter has passed. When the chosen day arrives, the root is still connected to a living crown of leaves. On a small farm or in a garden, a hand gathers the foliage near the base and pulls upward. The soil loosens around the root. Fine side roots tear free. For the first time since germination, the carrot's full surface enters daylight. Large farms use machines built around the same basic motion. A harvester lifts the leafy tops between moving belts. Metal blades or lifting shares loosen the soil below. The belts carry the plants upward while clumps of earth fall away. Some machines remove the foliage in the field. Others deliver whole carrots to a packing line where they are trimmed, washed, cooled, and sorted. The harvested root appears complete because it has reached the stage people want. Inside the plant's natural life cycle, it remains halfway through its work. During the first growing season, a carrot usually forms a low rosette of leaves and an enlarged storage root. The sugars held below ground were accumulated for the plant's own future. Those reserves could support rapid growth after winter, when a tall stem and hundreds of flowers would require more energy than a newly awakened leaf canopy could immediately provide. People harvest before that stored supply is used. At this point, the root is often sweet, crisp, and dense with living storage tissue. Once flowering begins, carbohydrates start moving away from the root. The crown produces a rising stalk. New leaves unfold higher above the ground. Flower clusters begin to form at the ends of branches. As those structures develop, the root may become firmer, less pleasant to eat, and increasingly fibrous. Fiber gives the plant stronger support for the work ahead, though it reduces the tender texture expected from a vegetable. Harvest therefore falls within a narrow biological opportunity. The plant has gathered a large reserve. Reproduction has not yet drawn heavily upon it. After lifting, the leaves are often removed quickly. A leafy top continues releasing water through its stomata. Moisture can move out of the root and into the foliage, then escape into the air. This causes the carrot to soften sooner. Leaves also take up space during packing and can carry soil or microbes from the field. Storage carrots are usually topped close to the crown, cooled, and held under very humid conditions. Mature topped roots can remain usable for several months when stored near 0 degrees Celsius with almost saturated humidity. Carrots sold in bunches with their green tops attached have a much shorter life. Even under good refrigeration, they generally keep for only about 10 to 14 days. Their leaves continue losing moisture, and the entire bunch wilts more quickly. Most harvested carrots now travel toward a familiar destination. Some enter grocery stores. Others become juice, frozen slices, soup ingredients, or the rounded pieces sold as baby carrots. A small number follow a different path. Seed producers save selected plants so the life cycle can continue. They may choose roots with a desired color, shape, size, or resistance to disease. Weak plants are discarded. Roots that split, rot, or differ from the intended variety may also be removed. The selected carrots can remain in the field where winters are mild enough. In colder or less predictable regions, growers may lift them, trim the foliage, store them under controlled conditions, and replant them later. Picture one carrot left behind after the harvest crew has moved on. Its neighbors have been pulled from the row. The remaining leaves sit low against the cooling ground. Below them, the root still holds months of stored carbohydrate. Those reserves are ready to support a second season, though the plant has not yet received permission to begin. Passing days alone will not trigger the change. The carrot must experience a long enough period of cold, allowing winter itself to become the signal that flowering may safely follow. Cold settles over the field, and growth slows almost to stillness. The low rosette may remain partly green in a mild winter. Under harsher conditions, many leaves yellow, collapse, or disappear close to the crown. Below the surface, the storage root remains alive. Its cells continue respiring at a reduced rate, using small amounts of the carbohydrate gathered during summer. Soil protects the root from sudden swings in air temperature. A few centimeters underground, conditions often change more slowly than they do above the surface. Seed growers can also remove selected carrots from the field before severe freezing arrives. The tops are trimmed, leaving a short crown. Roots are placed in cool storage where temperature and humidity can be controlled. During this cold period, the carrot undergoes a change called vernalization. Vernalization is the effect of prolonged cold on a plant's later ability to flower. The root does not measure winter with a calendar. Living cells near the growing point respond to weeks of low temperature. Cold alters the activity of genes that control whether the shoot keeps producing leaves or begins forming a flowering stem. For reliable flower initiation, agricultural guidance often recommends about 6 to 8 weeks between 2 and 5 degrees Celsius. That temperature range is cold enough to provide a strong seasonal signal, while remaining above the point where stored roots are likely to freeze solid. Field vernalization can work as well. In suitable climates, carrots may remain outdoors through at least 10 weeks, below 15 degrees Celsius. The roots must survive without being killed by deep frost or prolonged waterlogging. A protective layer of soil, straw, or snow can reduce temperature extremes. Two main seed production systems use this cold requirement in different ways. Seed-to-seed production leaves the plants where they first grew. The carrots pass through winter in the field and begin their second season in the same soil. This method requires less handling and can work well where winters remain cold enough for vernalization without becoming deadly. Root-to-seed production includes a deliberate pause. Growers lift the carrots after their first season and examine each one. The foliage is cut back. Selected roots are placed in refrigeration or another cool storage space. When spring conditions become suitable, those roots are replanted in rows prepared for seed production. Lifting the crop allows close inspection. A breeder can reject roots with rot, cracks, insect tunnels, weak crowns, or shapes that differ from the desired variety. An orange cultivar may be checked for uneven color or a pale core. A long-rooted type may be judged for taper, length, and straightness. Only the plants that best match the breeding goal are allowed to flower and pass their genes forward. Storage also reveals weaknesses that were less obvious at harvest. A root carrying hidden disease may soften or decay during the cold period. One with poor structure may shrivel more quickly than its neighbors. Removing such plants helps protect the seed field and preserves the chosen traits. After replanting, warmth and longer spring days allow the stored signal to take effect. The crown becomes active. New leaves appear, and the central growing point changes its pattern. Seed stalk development may become visible within about 4 to 6 weeks. This timing creates a risk during ordinary carrot production. Young plants sometimes experience several weeks of unexpected cold early in their first season. Warm weather then returns. The plant may interpret that sequence as winter followed by spring. A flowering stalk rises before the storage root reaches the size or texture growers wanted. This early shift is called bolting. The root often becomes tougher as energy moves toward the stem and developing flowers. Cultivars differ in how easily they bolt, which is why breeders select plants suited to local planting seasons and temperature patterns. Cold has now changed the future of the remaining carrot. The root still holds the reserves collected during its first year. while the growing point has gained the ability to use them for reproduction. As spring warms the soil, the compact rosette begins changing shape. A central stalk starts lifting the plant above the ground. Within a few weeks, the compact plant begins to look almost unfamiliar. The central stem lengthens above the low leaves and divides into branches. A mature flowering carrot can reach close to 1 meter in height, depending on the cultivar and growing conditions. Stored carbohydrates move upward from the root and help build this sudden column of new tissue. The stem must support leaves, transport water, and lift the flowers where insects can reach them easily. At the end of each branch, many thin flower stalks spread from a shared point. The shape resembles the ribs of an open umbrella. This structure is called an umbel. Carrots belong to the Apiaceae family, a name linked to these broad, umbrella-shaped flower clusters. Parsley, celery, fennel, and parsnip build similar arrangements. A carrot's umbel contains smaller groups called umbelles. Each umbellet carries numerous tiny flowers. Viewed from a distance, the whole cluster may look like one white bloom. Up close, it becomes a crowded platform made from many separate flowers, each only a few millimeters across. The flowers usually have 5 small petals. Their open shape makes nectar and pollen easy to reach. Many kinds of insects can land on the flat surface. Small flies often crawl across several flowers during one visit. Bees collect nectar and pollen, while beetles may feed and move between nearby umbels. As their bodies brush against the flower parts, pollen grains attach to hairs, legs, or mouth parts. A later visit can carry those grains to another flower. Pollen contains the male reproductive cells of the plant. When a compatible grain reaches the receptive surface of a flower, it grows a narrow tube downward. The male cells travel through that tube to the ovule, where fertilization takes place. The ovule then develops into a seed containing a new embryo. Seed growers depend on this insect movement, though they often manage it carefully. A field may receive naturally occurring pollinators from hedgerows, roadsides, and nearby meadows. Managed honeybee colonies can also be placed close to the crop. Some producers release flies because carrot flowers are especially attractive to many fly species. The flat umbels provide wide landing surfaces, and the shallow flowers suit insects with relatively short mouthparts. Cross-pollination creates an important challenge. Cultivated carrots can exchange pollen with other carrot varieties and with wild carrot growing nearby. A seed field containing a carefully selected orange cultivar may therefore be separated by a large distance from incompatible plants. The exact isolation distance depends on local rules, landscape, and seed class. Wind does not carry most carrot pollen efficiently over long distances, though insects can travel far enough to mix neighboring populations. Wild carrot pollen can introduce traits that growers do not want. The resulting offspring may develop paler roots, rougher texture, weaker color, or shapes closer to the thin wild form. Seed producers remove unwanted flowering plants from fields and nearby borders when possible. After successful pollination, the petals fade. The base of each flower begins swelling into a dry fruit. Over roughly 4 to 6 weeks, the developing fruits change from green to tan and then brown. The once flat umbel may curve inward as it dries. Changes in humidity can make the branches open and close slightly. helping mature fruits loosen and spread. Each dry fruit is a schizocarp. As it matures, it separates into 2 mericarps. Each mericarp contains 1 true seed and carries the familiar ribs and bristles seen at the beginning of the plant's life. Harvested seed heads are dried carefully before threshing, which separates the mericarps from stems and other plant material. Carrot seed has little deep dormancy. Once it is mature, dry, and placed under suitable conditions, it can germinate without waiting through another long seasonal cycle. The embryo inside already contains the beginning of a new root and shoot. The life cycle has now returned to a structure only a few millimeters long. Yet the flowering plant standing above the soil resembles a wild roadside carrot, far more closely than the thick orange vegetable found in a kitchen. That difference was built gradually, through many generations of people choosing which roots would be allowed to flower and which seeds would be planted again. Place a cultivated carrot beside a wild one, and their relationship becomes clear above the ground. Both plants form finely divided leaves with a soft, feathery look. During the second year, each can raise a branching stem topped with broad white umbels. Their flowers attract many of the same insects. Below the soil, however, the contrast is much greater. The cultivated plant may carry a thick root 20 centimeters long, brightly colored and rich in soft storage tissue. A wild carrot usually produces a narrow, pale root that may resemble a tough cord. Its surface can be rough, and its inner tissue often becomes woody as the plant ages. Crushing or scratching the root can release a familiar carrot scent. The smell reveals the family connection more clearly than the shape does. Wild carrot is known scientifically as Dorcas carota. It follows the same general 2-year rhythm as the crop. During its first season, it forms a low rosette and a thin taproot. Cold weather prepares the growing point for flowering. A tall stalk rises during the second season and may reach about 150 centimeters, close to the height of an adult person's shoulder. The main flower clusters can spread roughly 7 centimeters across. Dozens of narrow stalks radiate outward, carrying many small white flowers. Some umbels have a single dark red or purple floret near the center. This tiny mark is sometimes mistaken for an insect from a distance. Its exact purpose remains uncertain, though it may help draw the attention of visiting insects. The native range of wild carrot covers a broad part of the Old World. Populations occur in Macaronesia, the Atlantic island groups that include Madeira and the Canary Islands. The species also grows across parts of North Africa. Its range extends through much of Europe and Western Asia, reaching into regions of southern China. Within this enormous area, different populations have adapted to local winters, rainfall patterns, soils, and day lengths. Humans later carried wild carrot far beyond its original range. It now grows widely in North America, where it is often called Queen Anne's lace. Roadsides, abandoned fields, railway edges, and open grasslands can provide suitable habitat. The plant does especially well where soil has been disturbed and taller vegetation has not yet closed over the space. Its flowers support a varied community of visitors. The shallow structure allows insects with short mouthparts to reach nectar easily. Small bees move between neighboring flowers. Hoverflies may rest on the broad surface while feeding. Beetles can crawl through the cluster and carry pollen on their bodies. One flowering plant may produce around 1,000 fruits under limited conditions. Large, vigorous individuals have been reported with totals approaching 40,000. As the fruits dry, their hooked bristles catch on fur, feathers, socks, or other passing surfaces. Some simply fall beside the parent plant. Others travel to a new patch of open ground. The wild form belongs to a family that demands careful identification. Apiaceae includes familiar foods, such as parsley and celery. It also includes poison hemlock, Conium maculatum, and water hemlocks in the genus Cicuta. These plants can share white umbrella-shaped flower clusters and divided leaves. Several contain powerful toxins. Broad resemblance within the family provides too little information for safe foraging. Leaf shape, stem markings, smell, habitat, fruit structure, and several other traits must be considered by trained experts. The wild carrot's ecological success comes from traits that differ from those valued in a kitchen. A thin, tough root costs less energy to build than a large, tender one. Numerous small fruits improve the chance that some offspring reach open soil. Strong aromas and defensive chemicals may help protect living tissues from feeding animals or microbes. The cultivated carrot still carries much of this older biology. Its umbel's 2-year life cycle, aromatic compounds, and ability to cross with wild populations all preserve the connection. The thick storage root represents a later layer added through human choice. Generation after generation, growers saved seed from plants whose underground parts were larger, smoother, less woody, or more pleasant to eat. Small preferences accumulated until the root began to look very different from its wild ancestor. Across Western and Central Asia, farmers gradually changed the wild carrot into a crop. The process intensified during the early Middle Ages, especially around the 9th and 10th centuries. No single field or village can claim the moment when domestication began. Useful traits accumulated across a wide region over many planting seasons. People in this area had already known wild carrots for a long time. The aromatic leaves and fruits could be gathered for flavor, medicine, or scent. The thin roots also carried the familiar carrot smell, even though they offered little soft flesh. Cultivation created a new opportunity. Once plants grew close together in managed soil, farmers could compare them directly. Some roots were slightly thicker. Others grew straighter or contained less woody tissue. A few tasted milder, with less bitterness, and a more useful amount of stored food. Those differences came partly from the genes each plant inherited. A farmer who wanted more roots of a certain kind had to let selected plants survive into their second year. The chosen carrots passed through winter, produced tall flower stalks, and opened white umbels. Seeds collected from those plants carried shuffled combinations of traits into the following generation. This practice is called artificial selection. The word artificial refers to human choice, guiding which plants reproduce. Variation already exists inside the population. Selection changes how common each inherited trait becomes over time. Imagine a group of wild-looking carrots with mostly thin roots. One plant develops a root that is slightly broader and less fibrous. Its seeds are saved while seeds from poorer roots are ignored. Among the offspring, several may inherit part of that useful tendency. The best roots are chosen again. After many cycles, thick roots become less unusual. The average population slowly shifts. Several traits could be selected at the same time. A useful root needed enough size to justify digging it from the soil. Straight growth made harvesting and cleaning easier. Softer tissue improved cooking. Reduced bitterness made the crop more pleasant to eat. Strong color may have helped farmers recognize and preserve particular types. The earliest domesticated carrots probably looked very different from the standard orange roots common in Europe and North America today. Historical evidence and modern genetics point toward important purple and yellow forms in early cultivation. Purple roots accumulated pigments called anthocyanins. These are the same broad family of compounds that color blackberries, red cabbage, and many dark grapes. Yellow carrots contained a different balance of pigments, including yellow carotenoids. Orange would become dominant much later in Western Europe. Root appearance was only one part of domestication. Flowering time also mattered. A carrot that bolted during its first growing season sent energy into a flower stalk before producing a valuable storage root. Farmers favored plants that remained in the leafy stage long enough to build useful underground reserves. Local winters created further pressure. A population grown in one region might receive enough cold to flower reliably. The same plants moved into another climate could bolt too early or fail to produce seed at the right time. Day length added another signal. Plants use internal timing systems to track daily cycles of light and darkness. These systems are often described as circadian clocks. The word circadian means a rhythm lasting roughly 24 hours. Genes connected with these rhythms help coordinate leaf movement, growth, and flowering responses. Modern genetic research has made this long history easier to trace. In 2023, scientists studied an improved carrot reference genome and compared 630 accessions. An accession is a stored sample, representing a variety, landrace, breeding line, or wild population. The researchers found patterns linking domestication with changes in root traits, pigments, flowering control, and circadian genes. Their results placed the main early domestication process across Western to Central Asia during the early Middle Ages. The same work also showed a gradual loss of genetic diversity. When farmers repeatedly choose a smaller group of preferred plants, fewer genetic variants enter future generations. Later crop improvement narrowed that diversity further. The result was a more dependable vegetable with larger roots and recognizable traits. It also carried a smaller sample of the variation found across wild populations. By the end of this early domestication period, the carrot had become a true root crop. Its seeds could now travel with farmers and merchants into new soils, climates, and cooking traditions. Each region would continue the work, changing the crop again as it moved east and west. Seeds from the early cultivated populations began traveling far beyond Western and Central Asia. They moved in small cloth bags, clay jars, and bundles carried beside other useful crops. Farmers brought them into new fields. Merchants passed them along trade routes. Military campaigns, migration, and household gardens carried the plants across borders that would change many times over the centuries. Each journey moved more than a vegetable. It also moved local knowledge about planting, cooking, seed saving, and medicine. Some carrot populations traveled west through Persia, the Middle East, and North Africa toward Europe. Others spread east into South Asia and farther across the continent. These routes crossed deserts, river valleys, mountain passes, and regions with very different winters. A carrot suited to one place could behave poorly in another. Day length changes with latitude. Farther from the equator, summer days become longer and seasonal differences become stronger. Carrots use these patterns along with temperature to control growth and flowering. A population that received the wrong combination of cold and daylight could bolt too early. Its central stalk would rise before the root had stored enough food. For growers interested in the root, that plant offered little value. Seed from better time plants was more likely to be saved. Winter created another filter. Some regions had long, cold seasons that provided reliable vernalization. Others had mild winters where flowering signals were weaker. Local populations gradually developed different cold requirements. Soil also shaped selection. Long carrots perform best in deep, loose ground. Shorter or wider roots could succeed where soil was shallow, heavy, or filled with stones. Disease pressure varied between humid valleys and dry inland fields. Insects present in one region might be absent from another. Over many generations, each farming community kept the plants that worked best under its own conditions. Color traditions developed along these routes. Purple and yellow carrots remained important across parts of Asia. Deep purple roots were valued in several regional cuisines and could release dark pigment into soups, rice dishes, or pickles. Yellow types offered a milder appearance and a different balance of carotenoids. White and pale forms also persisted. Orange carrots would later become strongly associated with Western Europe, though they represented only one branch of a much wider cultivated family. Shape reflected local use. A long root could be sliced into strips or stored whole. Broad roots provided more mass from a shorter section of soil. Some populations were selected for tenderness when cooked. Others needed to survive long storage after harvest. Strong flavor might be welcomed in one dish and rejected in another. Seed saving continued to adjust the crop without any central breeding program. The carrot also had uses beyond its root. Aromatic fruits were sometimes collected for flavoring or traditional remedies. Leaves could be used when young, though they were usually less important than the roots in established vegetable crops. The strong scent of crushed tissue linked cultivated forms to wild carrot and other members of the parsley family. Different communities preserved a population because it fit a particular meal, climate, growing season, or custom. Reconstructing these movements is difficult. Medieval writers did not always use plant names with modern precision. One word could refer to carrot, parsnip, or another pale root. Descriptions might focus on flavor or medicine, while saying little about color. Painted manuscripts provide clues, though pigments can fade over hundreds of years. An artist might simplify a root or choose a color for visual balance rather than botanical accuracy. A painted orange vegetable does not automatically prove the existence of a modern orange cultivar. Written records and artwork become more useful when combined with genetics. DNA comparisons can reveal shared ancestry between regional populations. They can also show where groups separated, mixed again, or passed through periods when only a small number of plants contributed to later generations. This evidence supports a broad movement from Western and Central Asian domesticated stocks into both European and Asian traditions. Crossbreeding continued wherever different populations met. Pollen carried by insects could mix nearby cultivars. Farmers could also plant seed obtained from distant markets beside local varieties. The resulting offspring carried new combinations of root shape, pigment, and flowering time. The carrot became a traveling crop shaped by many hands and many landscapes. Its diversity expanded across the regions where people continued choosing which plants would reproduce. Among all these colors and forms, orange roots eventually gained unusual influence in Western Europe. Their rise was gradual, though their success became so complete that later generations often assumed carrots had always looked that way. A market in late medieval Europe would have offered carrots in several colors. Purple roots were familiar. Yellow types remained common. Pale carrots and roots with mixed coloring also appeared in fields and kitchens. Orange had not yet become the standard image attached to the word carrot. That change gathered strength during the Renaissance, the period of European cultural and scientific growth that stretched roughly from the 14th through the 17th centuries. In Western Europe, growers began saving seed from carrots with stronger orange color. These roots contained high levels of carotenoids, a family of yellow, orange, and red pigments made by plants. Beta-carotene was especially important, though farmers selecting the roots had no way to see individual pigment molecules. They worked with qualities they could observe directly. A bright root stood out clearly after harvest. Even color made a crop look more consistent at market. Certain orange populations may also have offered useful flavor, texture, shape, or storage qualities. When several desirable traits occurred in the same plant, growers had a strong reason to preserve its seed. Color could deepen across generations through repeated selection. A carrot carrying genetic variants that increased carotenoid production might appear slightly more orange than the others around it. Allowing that plant to flower gave those variants another chance to enter the following crop. Further selection concentrated useful combinations. Over time, orange roots became more frequent, more intense in color, and more dependable from one planting to the next. Dutch growers played an important part in improving and spreading these carrots. The Netherlands became highly skilled in vegetable cultivation and seed production during the early modern period. Dutch seed growers helped develop stable orange varieties that could be distributed more widely across Europe. A popular story connects this work directly to the House of Orange, the royal family associated with Dutch independence. According to the legend, growers created orange carrots as a patriotic tribute to William of Orange. The historical record supports a slower and less tidy account. Orange carrots developed through generations of breeding, with contributions from growers across Western Europe. Dutch agriculture helped refine and popularize them. No surviving document records one planned moment when farmers decided to invent the orange carrot for a royal family. The political story remained popular because it was simple, colorful, and easy to remember. DNA preserves a more detailed record. In 2023, researchers compared the genomes of 630 carrot accessions, including wild plants, regional varieties, and modern breeding lines. A genome is the complete set of genetic instructions carried by an organism. The study found evidence of strong selection for orange roots during the Renaissance, probably in Western Europe. 3 genes showed especially strong links with orange pigmentation. Changes around these genes helped alter how carotenoids were produced, stored, or broken down inside root cells. The result was a much greater concentration of orange pigment. Selection also affected genes connected with flowering time and daily biological rhythms. A successful orange carrot still needed to remain in its first-year growth stage long enough to build a large root. Beautiful color offered little value if the plant sent up a flower stalk too early. Breeders therefore preserved combinations that joined strong pigmentation with useful timing and dependable root growth. This improvement left another mark in the genome. Early domestication had already reduced genetic diversity because only part of the wild population contributed heavily to the crop. Later breeding narrowed the gene pool again. This kind of reduction is called a genetic bottleneck. Imagine a wide river passing through a narrow channel. Only some of the original variation makes it through. Strong selection for orange color, root shape, and flowering behavior created an additional bottleneck as preferred plants produced a larger share of future seed. By the 17th and 18th centuries, orange carrots were becoming firmly established across Western European agriculture. Later trade and colonization carried them into many other regions. Their consistency suited expanding markets, while their color became linked with the crop itself. Centuries after the first purple and yellow cultivated roots appeared in Asia, one later branch came to represent the whole family. The familiar orange surface now carries a record of Renaissance farming, Dutch seed production, and repeated genetic selection. Its color is produced by a particular set of pigment pathways. Other carrots follow different chemical routes, filling their roots with purple anthocyanins, yellow xanthophylls, red lycopene, or very little visible pigment at all. Across the full cultivated range, carrots appear in white, yellow, orange, red, purple, and shades so dark they can look almost black. Each color comes from a different balance of pigments inside the root. These pigments are molecules that absorb certain wavelengths of light and reflect others back to the eye. A purple carrot contains different chemistry from an orange one. A red carrot follows another pathway again. The colors do not form a simple ladder from pale to dark. They are separate biological outcomes created by different genes and enzymes. Orange carrots build up large amounts of carotenoids. Carotenoids are fat-soluble pigments found in many plants. They help produce the color of pumpkins, sweet potatoes, and autumn leaves. Carrot roots commonly store alpha-carotene and beta-carotene. Both absorb much of the blue and green light that reaches them, leaving orange wavelengths to be reflected. Beta-carotene often receives more attention because the human body can use it to make vitamin A. Yellow carrots usually contain a greater share of pigments called xanthophylls. Xanthophylls belong to the wider carotenoid family. Lutein is one familiar example. It also occurs in leafy greens, corn, and egg yolks. A yellow carrot may contain some beta-carotene, though its pigment balance gives the root a softer golden color. Red carrots can accumulate lycopene. This pigment is also responsible for much of the red color in ripe tomatoes and watermelon. Lycopene absorbs light differently from orange carotenoids, producing a deeper red tone. Some red cultivars also contain other carotenoids, so their color may range from coral to dark crimson. Purple and nearly black carrots rely heavily on anthocyanins. Anthocyanins are water-soluble pigments found in blueberries, blackberries, and red cabbage. Many purple carrots contain pigments based on a molecule called cyanidin. These compounds may appear red, violet, or blue, depending on their exact structure and the chemical conditions inside the cells. A very dark carrot can contain such a high concentration that the surface looks almost black. White carrots contain relatively little strongly colored carotenoid or anthocyanin pigment. Their tissues still hold sugars, cell walls, proteins, and aromatic chemicals. The lack of strong color comes from low pigment accumulation rather than an empty or inactive root. Pigment may also vary within one carrot. Slice a purple cultivar across the middle, and the outer ring may appear dark while the center remains yellow or orange. The outer region contains more phloem and storage tissue. The inner core contains a greater share of xylem. Genes can control whether anthocyanins collect mainly in the outer phloem, throughout the inner xylem, or across both regions. This creates roots with rings, dark skins, or sharply contrasting centers. The pattern develops as enzymes turn simple starting materials into pigment molecules. A biosynthetic pathway is the step-by-step chemical route used to build a compound. Each step depends on an enzyme, and each enzyme is produced according to genetic instructions. A change in one control gene can increase pigment production across the whole route. Another change may limit color to one tissue layer. Breeders select these visible patterns by saving seed from plants with the desired result. Pigments also serve the plant. Carotenoids help chloroplasts manage light energy in green tissues. Anthocyanins can absorb intense light and take part in responses to cold, injury, and other forms of stress. Several pigments help control reactive oxygen molecules. These unstable molecules form during normal metabolism and can damage cells when they build up too quickly. The root usually grows in darkness, though its pigment pathways remain part of the wider chemistry shared across the plant. Color can still influence protection after injury, or when the top of a root becomes exposed near the soil surface. Humans later gave these pigments meanings far beyond their role inside the plant. Purple roots became linked with traditional dishes in parts of Asia. Orange became the expected market color across much of Europe and North America. Beta-carotene gained a special reputation because digestion can carry it from a root into the human body. There, enzymes can convert it into compounds used by light-sensitive cells inside the eye. Once a carrot is chewed, its firm tissues begin to break apart. Teeth crush the storage cells, and saliva moistens the pieces before they move through the stomach. The pigments remain tucked inside oily parts of the food until digestion reaches the small intestine. There, bile from the liver helps break dietary fat into tiny droplets. Carotenoids can join these droplets and move close to the intestinal wall, where some of them are absorbed. A small amount of fat in the meal can improve this process. because carotenoids dissolve more easily in fat than in water. Beta-carotene belongs to a group called provitamin A carotenoids. The word provitamin means that the body can use the compound as raw material for making a vitamin. Alpha-carotene and beta-cryptoxanthin can also serve this role. Beta-carotene itself is still a plant pigment when it enters the intestine. An enzyme called beta-carotene monooxygenase 1 can split the molecule near its center. The pieces are then changed into retinoids, the family of compounds related to vitamin A. Conversion efficiency differs from one person to another. Genes influence how much of the enzyme a person produces and how actively it works. Digestive health, food preparation, and the amount of fat in the meal also matter. A cooked or finely chopped carrot may release more carotenoid than a large raw piece because heat and cutting weaken the plant's cell structure. The body also adjusts conversion according to its needs. When vitamin A stores are sufficient, less beta-carotene may be changed. This control makes food-based carotenoids different from large amounts of preformed vitamin A taken directly from certain animal foods or supplements. Vitamin A supports several systems throughout the body. It helps guide normal growth and keeps surface tissues healthy. Immune cells depend on it as they respond to infection. Developing embryos also require carefully controlled amounts. Its most famous role takes place in the retina, the thin layer of light-sensitive tissue lining the back of the eye. The retina contains two main kinds of light-detecting cells. Cone cells work best in brighter light and support detailed color vision. Rod cells are much more sensitive in dim conditions. A human retina contains roughly 120 million rods, though the exact number varies. Each rod is packed with a light-sensitive pigment called rhodopsin. Rhodopsin forms when a vitamin A-derived molecule called retinol joins with a protein called opsin. When light strikes retinol, the molecule changes shape. That tiny change starts a chain of electrical activity inside the rod cell. Signals then pass through other retinal cells and travel along the optic nerve toward the brain. After retinol changes shape, the visual system must rebuild the light-sensitive form before the molecule can respond again. This recycling process continues constantly. Moving from a bright room into darkness reveals the time it takes. At first, the room may seem almost black. Over the following minutes, rod cells restore more rhodopsin and become increasingly sensitive. Full dark adaptation can take around 20 to 30 minutes. A severe shortage of vitamin A slows this system. Too little retinol means the rods cannot maintain enough rhodopsin. One early result can be night blindness, a reduced ability to see in dim light, or adjust after brightness. In regions where vitamin A deficiency is common, foods rich in provitamin A can help protect normal vision. Carrots may contribute because their beta-carotene provides material the body can convert. Adequate vitamin A supports the eye's normal machinery. Extra carrots cannot reshape the cornea or change the length of the eyeball. Nearsightedness occurs when incoming light focuses in front of the retina, often because the eye is too long or its focusing surfaces bend light too strongly. Farsightedness and astigmatism also involve the physical path of light through the eye. Glasses, contact lenses, or medical care address those conditions far more directly than diet. Eating very large amounts of beta-carotene-rich food can sometimes tint the skin yellow-orange. This harmless condition is called carotenodermia. The color often appears first on the palms or soles and gradually fades after intake returns to ordinary levels. The whites of the eyes remain clear, which helps distinguish it from jaundice. Carrots therefore have a real connection to vision, though the connection follows a precise biological path. Pigment leaves the root, passes through digestion, becomes a retinoid, and joins the light-sensitive chemistry of the retina. That modest truth would later grow into a much larger story during the Second World War, when British cities dimmed their lights and darkness became part of daily life. Britain began preparing for darkness before German bombs fell in large numbers. On September 1st, 1939, 2 days before Britain declared war on Germany, blackout rules came into force across the country. Street lamps were switched off. Homes covered windows with thick curtains or dark paper. Vehicle headlights were fitted with narrow masks that allowed only a small amount of light to reach the road. The goal was to make towns, railways, and factories harder for enemy aircrews to locate at night. Ordinary movement became more difficult. Pedestrians crossed streets with fewer visual clues. Drivers followed dim strips of road. Accidents increased as millions of people adjusted to cities that disappeared after sunset. The blackout became especially important during the Blitz, the bombing campaign that struck London and other British cities from September 1940 until May 1941. At the same time, food supplies were under pressure. German submarines attacked ships carrying food across the Atlantic. Britain imported much of what its population ate, so the government encouraged farmers and households to produce more food at home. The Ministry of Food taught people how to use crops that could be grown in British soil. Carrots were useful because they stored well, grew in cool weather, and could be added to soups or stews. They also brought natural sweetness to recipes when imported sugar was tightly rationed. Government campaigns promoted carrot cakes, puddings, drinks, and savory dishes. One surviving Ministry of Food poster measures 758 by 508 millimeters, a little taller than a large kitchen cupboard door. Its message reads, "Carrots keep you healthy and help you to see in the blackout." The claim rested on a real nutritional connection. Carrots provide beta-carotene, which the body can convert into vitamin A. A person lacking enough vitamin A may struggle to see in dim conditions. Restoring that missing nutrient can improve normal night vision. Most well-nourished people already have enough vitamin A to maintain the light-sensitive chemistry inside their eyes. Extra carrots do not give healthy eyes a new level of power. Wartime conditions gave the message unusual strength. The public was living under darkness rules every evening. Carrots were available during a period of shortage. An official poster linked the two subjects with a simple promise that was easy to remember. Stories about British night fighter pilots added another layer. Newspapers and public campaigns sometimes suggested that successful pilots owed their sharp night vision to eating carrots. The Royal Air Force was indeed becoming better at finding enemy aircraft in darkness. That success depended on a developing network of radar stations, ground controllers, specialized aircraft, and trained crews. Radar sent out radio waves and detected echoes returning from distant objects. Ground stations use that information to help guide fighters toward approaching bombers. Aircraft later carried their own compact interception radar, allowing crews to search through darkness and cloud. The technology was secret, and Britain had good reason to avoid explaining its full abilities. A popular later story claims that the carrot message was created as a planned deception to hide radar from Germany. The documented record supports a genuine food campaign and repeated claims about carrots helping with blackout vision. Evidence for one carefully organized deception behind the entire campaign remains much less complete. Several motives could exist at the same time. Officials wanted people to eat available crops. They wanted to improve nutrition during rationing. They also preferred that enemy forces remain uncertain about British detection systems. Over the years, these pieces blended into a cleaner legend. According to the familiar version, pilots ate carrots, gained remarkable night vision, and surprised enemy aircraft in darkness. The real history contains more moving parts. Nutrition supplied the small truth at the center. Wartime secrecy gave the story mystery. Official repetition helped it spread far beyond the years of bombing. The myth endured because it attached a complicated subject to an ordinary vegetable. Radar physics and military coordination were difficult to explain in a short poster. A bright orange root offered a picture that anyone could remember. Its reputation for eyesight survived long after blackout curtains came down. Yet vision reveals only one part of the carrot's chemistry. The experience of eating one depends on sugars, aromatic oils, bitter compounds, and the way those molecules reach both the tongue and the nose. The first bite begins with sound. A raw carrot breaks with a sharp snap, as tightly packed cells and stiff cell walls give way. Juice spreads across the tongue almost at once. Sweetness appears first in many varieties, followed by the greener, earthier notes that make the flavor recognizable. Some carrots finish with a faint bitterness. Others leave a dry feeling along the sides of the mouth. Those differences come from many compounds arriving together. The main free sugars in carrot roots are glucose, fructose, and sucrose. Glucose and fructose are simple sugars. Sucrose is made from one unit of each joined together. Their amounts change with cultivar, age, growing conditions, and storage. A carrot with more sugar often tastes sweeter, though sugar alone cannot predict whether people will enjoy it. The familiar carrot aroma comes largely from compounds called terpenes. Terpenes are small molecules produced by plants for scent, defense, and communication. They evaporate easily, which allows them to reach the nose. Some create fresh, herbal notes. Others smell pine-like or slightly earthy. A recent scientific review gathered reports of more than 200 volatile compounds in carrots. Volatile means that a substance can enter the air easily enough to be smelled. The exact mixture changes from one root to another. Compounds such as saponine, terpinolene, and beta-caryophyllene have been linked with parts of the characteristic aroma. Their names matter less than the balance between them. A tiny amount may add freshness. A higher concentration can make a carrot taste harsh or strongly medicinal. Bitterness comes from several chemical groups. Polyacetylenes are among the most important. One example is falcarindiol, a defensive compound found in carrot tissue. It can discourage insects or microbes, while also adding bitterness at higher levels. Phenylpropanoids, another group of plant chemicals, can contribute sharpness or a drying sensation called astringency. Astringency is the puckering feeling produced when certain compounds interact with proteins in saliva. Much of what people call taste actually reaches the brain through smell. While chewing, volatile molecules rise from the back of the mouth into the nasal cavity. This route is called retronasal smell. It differs from sniffing food through the nostrils before eating, though both routes stimulate the same olfactory system. A blocked nose demonstrates the effect clearly. Sweetness and bitterness remain noticeable on the tongue, while much of the carrot's detailed aroma seems to disappear. Texture shapes the experience as well. A fresh root with firm cells gives a clean snap and releases juice quickly. A wilted carrot bends more easily because its cells have lost water and internal pressure. The same sugars and aromas may still be present, yet the softer texture changes how the flavor is judged. Different tissues inside one root can also taste slightly different. The outer phloem-rich region often contains more sugars and pigments. The central xylem core may be firmer and less sweet in some cultivars. Breeders sometimes select for a small core and even flavor from edge to center. Weather influences the balance before harvest. Cool growing conditions can support slow, steady sugar accumulation. Heat and drought may strengthen bitter or harsh notes. Uneven watering changes growth rate and can alter texture. Damage from insects, cutting, or disease activates chemical defense pathways, causing the root to produce or release compounds that were less noticeable before injury. Storage continues the change. Carrots remain alive after harvest. They respire, lose moisture, and slowly use stored sugars. Long storage can weaken fresh aromas or allow bitter notes to become more obvious. Cold temperatures slow these processes, which is why commercial roots are cooled soon after harvest. Sensory scientists measure several qualities at once when comparing varieties. They score sweetness and bitterness, along with aroma intensity, texture, astringency, and unwanted flavors. A root with very high sugar may still be judged poorly if strong terpenes dominate. A mildly bitter carrot can taste balanced when sweetness and aroma soften the effect. Flavor emerges from proportion. The tongue detects dissolved compounds. The nose receives airborne molecules. Teeth and jaw measure firmness as the tissue breaks. Expectation adds another layer, since a bright orange root may seem sweeter before it is tasted. This chemical arrangement begins changing as soon as the root meets a knife, a grater, hot water, or the dry heat of an oven. A raw carrot resists the teeth with a clean, rigid snap. After enough time in hot water, the same root yields under a fork and can be pressed into a soft mash. This change begins inside the walls and membranes that once kept millions of plant cells firmly arranged. Carrot cells are surrounded by walls made largely from cellulose, hemicellulose, and pectin. Pectin also helps hold neighboring cells together in a thin layer called the middle lamella. Heat weakens this pectin-rich material. Cell membranes lose their organized structure, and water moves more freely through the tissue. As the bonds between neighboring cells loosen, the carrot becomes easier to bend, cut, and chew. The method of heating determines what happens next. During boiling, carrot pieces sit directly in hot water. Sugars, minerals, and other water-soluble compounds can move out of damaged cells and enter the liquid. Some aroma molecules escape with the steam. The root softens, while its flavor may become milder. When the cooking water remains part of a soup or stew, many of the compounds that left the carrot are still eaten rather than poured away. Steaming keeps the vegetable above the water. Hot vapor transfers energy into the tissue, yet there is less direct contact with liquid. This usually reduces the amount of soluble material washed out of the root. The carrot still softens as heat weakens its internal structure. Roasting creates a different environment. Dry oven air removes water from the surface. As moisture escapes, sugars and flavor compounds become more concentrated near the outside. The surface temperature can rise much higher than it would in boiling water, which remains close to 100 degrees Celsius under ordinary conditions. At these higher temperatures, new aromas begin to form. One important process is the Maillard reaction. It occurs when certain sugars react with amino compounds during heating. The reaction produces many flavor and aroma molecules associated with browned food. A roasted carrot may develop toasted, nutty, or slightly caramel-like notes that were absent from the raw root. Caramelization can also occur as sugars break down under strong heat, especially where the surface becomes dry. Cutting changes the chemistry before cooking even begins. A whole carrot keeps much of its internal structure intact. Slicing creates many exposed surfaces. Grating ruptures far more cells, releasing juice and bringing enzymes into contact with compounds that were previously separated. Pureeing reduces the tissue into very small particles. Each step makes the plant material easier for digestive fluids to reach. Carotenoids such as beta-carotene are stored inside cell structures and attached to membranes. The human digestive system cannot always free all of them from raw, intact tissue. Heating can weaken the surrounding plant matrix. The matrix is the physical network of cell walls, membranes, and other material holding the food together. Once that network softens, a larger share of the carotenoids may be released during digestion. Scientists describe this available share as bioaccessible. Greater bioaccessibility means that more of the pigment can leave the food and enter the digestive mixture, where absorption becomes possible. The total amount of beta-carotene has not suddenly increased. Some may even be lost through long heating, exposure to oxygen, or contact with light. The important change concerns how easily the remaining pigment can be reached. Dietary fat adds another part to this process. Carotenoids dissolve in fat more readily than in water. Inside the small intestine, bile helps form tiny mixed particles that can carry fat and fat-soluble compounds toward the intestinal wall. A carrot eaten with olive oil, yogurt, eggs, or another source of fat may therefore supply carotenoids more efficiently than the same carrot eaten alone. The amount of fat needed can be modest. Food preparation creates a series of trade-offs. Boiling can soften the root quickly while moving some flavor into the water. Roasting strengthens certain aromas and concentrates sweetness near the surface. Pureeing can increase access to pigments while removing the crisp texture of the raw vegetable. Each method reshapes the same living tissue into a different chemical and physical form. Beyond the kitchen, carrots must tolerate a much longer chain of handling. They are lifted by machines, washed under pressure, cooled close to freezing, packed by size, and carried across hundreds or thousands of kilometers. That journey has pushed breeders to shape the route for an entire industrial system. A commercial carrot begins with a decision about its final job. Some will be sold whole in fresh produce aisles. Others will be sliced for frozen meals or ground into juice. A large number will become the smooth, rounded pieces sold in bags as baby carrots. Different root shapes suit different soils, machines, and markets. Imperator carrots are long and narrow, often reaching 20 to 30 centimeters. They grow best in deep, loose soil, where the tip can extend without meeting hard layers or stones. Their straight shape and strong orange color make them common in fresh markets across North America. Nantes carrots are usually shorter and more cylindrical. Their sides remain fairly even before narrowing near the tip. Many have a tender texture and a relatively small core, which makes them useful for fresh eating and processing. Chantenay carrots are shorter and broader, with wide shoulders and a strong taper. They can perform better in shallower or heavier soils, where a very long root would struggle to develop. Breeders work with these basic forms while selecting for dozens of smaller traits. The leaves must remain strong enough for machines that lift carrots by their tops. The crown should resist breaking. The root needs enough strength to survive contact with soil, steel, water jets, conveyor belts, and sorting equipment. Commercial harvesters loosen the ground beneath the row. Belts grip the foliage and pull the roots upward. Rotating parts remove soil while the carrots travel through the machine. A brittle root may snap during lifting. A weak top may tear away before the root leaves the ground. Bent or heavily forked carrots can become trapped or damaged along the line. After harvest, water removes clinging soil. Cameras and sensors may sort roots by length, diameter, color, and visible defects. Human inspectors also look for cracks, rough surfaces, green shoulders, excessive hairiness, poor trimming, and signs of decay. Green shoulders develop when the upper root is exposed to light and begins producing chlorophyll. The color itself is harmless, though it lowers visual quality and may accompany stronger flavor. Bitterness, firmness, straightness, and taper all influence whether a root reaches the fresh market or enters processing. Many baby carrots begin as full-size mature roots. Growers use cultivars selected for long, fairly uniform bodies, with strong color and a small central core. At the processing plant, each carrot is cut into several short sections. The pieces move through equipment that peels and rounds their edges by abrasion. Abrasion means that rough surfaces rub away thin outer layers. The finished pieces are washed, cooled, and sealed in bags. Their size depends partly on the original root. A long, straight carrot produces more usable sections than a short or sharply tapered one. Fresh-cut carrots remain living tissue. Cutting damages cells and exposes wet surfaces where microbes can grow. The pieces also lose moisture more quickly than whole-topped roots. Under good refrigeration, fresh-cut carrots may keep for about 3 to 4 weeks. Immature whole roots often last around 4 to 6 weeks. Carrots sold in bunches with leaves attached usually remain in good condition for only 10 to 14 days. Mature topped carrots can last far longer. The best long-term storage conditions are close to 0 degrees Celsius, with 98 to 100% relative humidity. Relative humidity describes how much moisture the air holds, compared with the maximum it could hold at that temperature. Nearly saturated air slows water loss from the root. Cold temperatures reduce respiration and delay microbial growth. Under these controlled conditions, mature topped carrots may remain usable for roughly 7 to 9 months. That long life allows harvest from one season to supply markets well into another. The scale extends far beyond any single storage room. In 2020, the combined global crop of carrots and turnips exceeded 41 million tons. China produced about 44% of that total. Moving such a crop requires breeding programs, irrigation systems, field crews, harvest machinery, refrigerated warehouses, packing lines, transport networks, and processors. Waste must also be handled, including broken roots, peelings, rejected pieces, and spoiled produce. The ordinary carrot on a dinner plate sits at the end of a large coordinated system. Uniform roots move through that system more efficiently because machines can grip, cut, wash, and pack them in predictable ways. Uniformity also creates biological exposure. A vast field of similar plants offers the same tissues to every insect, fungus, nematode, and pathogen capable of using them. From above, a carrot field can look calm and even. Rows of green leaves may appear healthy while damage spreads through the hidden roots below. Much of the crop's value remains underground, beyond easy inspection. By the time a problem becomes visible in the foliage, the marketable root may already be scarred, forked, or partly decayed. One of the clearest examples is the carrot rust fly. The adult is a small dark fly, only a few millimeters long. Females place their eggs in cracks in the soil close to carrot plants. After hatching, pale larvae move toward the roots and begin feeding. Their tunnels often appear as narrow rust-brown lines, especially around the lower part of the carrot. The channels damage storage tissue and create openings where other microbes can enter. A root may look acceptable near the crown, while deeper sections contain winding scars. Depending on climate, carrot rust flies may complete between 1 and 3 generations in a year. Later generations can be especially damaging because they attack roots that have already grown large. Fine mesh placed over a crop can block adult flies from reaching the soil. Growers may also adjust planting dates, remove crop remains, or harvest before late-season larvae cause greater injury. Another attacker is much smaller. Root knot nematodes are microscopic roundworms that live in soil. Young nematodes enter the root near growing tissue and move between cells. Once inside, they release chemicals that change normal plant development. Several nearby cells enlarge and become feeding sites known as giant cells. The nematode draws nutrients from these altered tissues, while remaining protected within the root. Swollen knots, called galls, may appear around the feeding area. Damage near the tip can disturb the carrot's main downward growth. The finished root may become forked or covered in fine side roots. Severe infection slows the whole plant because water and minerals no longer move through the damaged tissue efficiently. Some threats enter through insects but spread inside the plant's transport system. Aster yellows is caused by a phytoplasma, an extremely small disease-causing organism related to bacteria. It lives in phloem, the tissue that carries sugars through the plant. The aster leafhopper, Macrostelis quadrilineatus, picks up the phytoplasma while feeding on an infected plant. During later feeding, the insect can introduce it into a healthy carrot. Affected leaves may turn yellow or develop twisted growth. The crown can produce too many shoots, creating a crowded, brush-like top. Below ground, the root may become hairy, bitter, and poorly shaped. Once a carrot is infected, no treatment removes the phytoplasma from its tissues. Growers reduce risk by controlling weeds that can carry the disease and by monitoring leafhopper movement into fields. Wet weather creates a different opening. The fungus Alternaria dauchi causes carrot leaf blight. Dark lesions form on older leaves and can spread until much of the foliage dies. A weakened canopy produces less sugar for the root, while brittle leaves can break during mechanical harvest. The fungus may survive in infected crop debris. It can also occur on or within seed, allowing the disease to begin in a new field before obvious symptoms appear. Rain and overhead irrigation move spores between leaves. Long periods of moisture give those spores enough time to germinate and enter the tissue. Cool, wet soil favors several Pythium species. These organisms belong to a group often called water molds. Some attack young roots and kill the main tip, leading to short or forked carrots. Others contribute to cavity spot. The disease produces sunken lesions that may reach about 2.5 centimeters across. Dark marks on the surface can make an otherwise firm root unsuitable for sale. Protection depends on several measures working together. Clean seed reduces the chance of introducing certain diseases. Crop rotation lowers the number of pests that build up when carrots return to the same ground too often. Good drainage removes excess water, while resistant cultivars provide useful inherited defenses. Field monitoring helps growers act before a problem spreads widely. Harvest timing can limit the period during which mature roots remain exposed. No single method covers every entry route. Some attackers arrive through foliage, while others wait in soil or travel inside insects. The narrow genetic base that makes a modern crop uniform may also leave fewer defensive traits available. Future protection may depend on variations still preserved in wild carrots, older regional varieties, unusual colors, and carefully stored seed. That remaining variation has already changed the way scientists understand plants. In the late 1950s, two researchers working separately, F.C. Stewart and Jakob Reinert, used carrot cells in experiments that became important in plant biology. They began with ordinary body cells taken from carrot tissue. These were somatic cells, meaning cells from the plant's regular tissues, rather than pollen, eggs, or seeds. Under carefully controlled laboratory conditions, some of those cells began dividing. They formed small embryo-like structures. Given the right nutrients and plant hormones, those structures could develop into complete carrot plants with roots, leaves, and flowers. This process became known as somatic embryogenesis. It showed that a mature plant cell can sometimes return to a more flexible state and rebuild an entire organism. The ability is called developmental plasticity. Plasticity means that a living system can change its path when conditions change. A carrot cell taken from one part of a root still carried the full genetic instructions needed to produce every other part of the plant. Decades later, researchers learned to read those instructions in far greater detail. In 2016, scientists published a major carrot reference genome. A genome is the complete set of DNA carried by an organism. The assembly covered about 421.5 million DNA base pairs. Base pairs are the chemical units that form the long code inside DNA. Most of that sequence was organized into 9 chromosome scale structures, matching the carrot's 9 chromosomes. Researchers predicted more than 32,000 genes. Some were linked with flowering time. Others helped control carotenoid pigments, root development, disease response, and the long history of crop improvement. The genome gave scientists a detailed map. Later studies could compare wild carrots, old regional types, modern cultivars, and breeding lines against the same reference. In 2023, researchers used that approach to study 630 carrot accessions. Each accession represented a preserved sample, such as a wild population, landrace, cultivar, or breeding line. The comparison helped reconstruct the crop's movement from early domestication in Western and Central Asia to later improvement in Europe. It also revealed the strong selection that produced orange carrots during the Renaissance. The same data showed repeated losses of genetic diversity. Domestication narrowed the range once. Modern breeding narrowed it again as growers favored uniform roots with dependable color, shape, and flowering behavior. Uniformity made farming easier. It also removed some inherited variation that may become useful under new conditions. That is why wild populations and older varieties still matter. A landrace is a locally adapted crop population, maintained by farmers over many generations. It may contain uneven shapes or colors, yet it can also carry useful traits related to heat, drought, soil, disease, or flowering time. Wild carrots preserve an even wider range of possibilities. One population may tolerate dry ground. Another may resist a local pest or flower under a different pattern of cold and daylight. No single trait will protect agriculture from every future problem. A larger genetic library gives breeders more material to study and combine. Seed banks help keep that library from disappearing. Dry seeds are cleaned, tested, labeled, and stored under cool conditions that slow aging. Kew's CropWild relative work has preserved more than 2.1 million wild carrot seeds. Those seeds represent living variation collected from different places and environments. Some may remain in storage for many years. Others are grown periodically, so fresh seed can replace aging supplies. Conservation also continues in fields and gardens. Farmers who maintain purple, yellow, red, or regional carrot types preserve combinations of genes that commercial markets might otherwise lose. Researchers can cross those plants with modern varieties, then select offspring that combine useful resistance with the color, flavor, or shape people expect. The process remains slow. A promising cross may require many growing seasons before its traits become stable. The carrot can now return to the hand where this journey began. Its water rose from soil through xylem. Its sugars traveled from leaves through phloem. The root widened through weeks of cell division, while stones, moisture, and temperature shaped its final form. Its color carries the work of enzymes guided by genes selected across centuries. Its sweetness came from carbon dioxide once floating in the air. The stored reserves were meant to power a second year, a tall flowering stem, and another generation of small dry fruits. Harvest stopped that cycle halfway. The root still holds the record of everything that happened before it was pulled from the ground. It carries one season of light, a winter signal it may never receive, more than 1,000 years of human choice, and a living collection of possibilities that agriculture may still need.